right. Hi, everybody. I want to review some simple genetic problems and terms. Understanding simple genetic problems and how they work is important for understanding inbreeding, and understanding inbreeding is an important part of conservation biology. So I'm gonna start off by showing you a cross between two individuals that are heterozygous for some particular trait. Now, as I do this, if you find that the terminology and the use of the terminology is overwhelming for you, I suggest you go back, or if you haven't, watch for a first time my Gregor Mendel video. I'll put the link down below. So let's say we're crossing, like I said, two heterozygous individuals that are carrying some recessive allele. And remember, an allele is an alternate form of some gene. And the letters that we pick or use here, N, capital N and lowercase n, those are really symbols to represent some piece of genetic code that represents a gene that it's going to code for. So we're going to cross two heterozygous individuals carrying a recessive allele for some genetic trait. And let's say the male is on the left over here, has the potential of producing two types of sperm. And one type of sperm has this capital N in it, and the other type has the lowercase n in it. And since animals have two sets of chromosomes in them, we have to sort of look at both. And so in this case, one of the chromosomes, maybe it's chromosome 4, has this capital N, and the other chromosome 4 has this lowercase n. So that's how we start. And here we drew these little tails on them to represent males and therefore sperm. But it doesn't really matter which ones you put where, as long as you have all the options, because this is more of a probability when we make this Punnett square. It's more of a probability of what could happen under different scenarios. So we have two types of sperm. In fact, 50% will be the dominant type, the capital N, and 50% will be the lowercase n, as you see there. And same thing for the female. The female in this particular example has the same setup in the sense that for that particular trait, she's also heterozygous and has a capital N, lowercase n. So the eggs will have one of each. So then what you do is you just ask yourself, well, what would happen if the uppercase or dominant N happened to fertilize the lowercase egg? What you would get is what we call a homozygous dominant individual. So it'd be phenotypically normal. Okay. So then what if the lowercase sperm or the recessive allele, the lowercase n, happened to fertilize the dominant N of the egg? Then you would have a heterozygous individual, and heterozygous means they have one of each type. They'd be phenotypically normal because in this particular scenario, we're talking about a genetic condition of some sort that's recessive, meaning that if you have one dominant allele, you don't see the effects of the recessive trait since it's sort of hidden by what the dominant allele can do. So then what if that sperm happened to fertilize that egg? You'd have a heterozygous cross once again, phenotypically normal. And then what if you had the lowercase n fertilize the lowercase egg? Well, if you did that, you would have a homozygous recessive baby or offspring. And what you would have in that scenario then would be the individual, that baby resulting from the offspring from that interaction would die of some particular genetic disease, assuming that the recessive trait caused a recessive allele expression that was harmful. So what are the chances of that happening? Well, based on that scenario there's a 25% chance, if you had those two individuals, of having a baby with that outcome. So that's the first part, 25%. If you had those two individuals come in contact, that's what you could expect to get. So the next question is, what's the probability of two individuals that are reproducing that have the same recessive allele issue for the same trait? Well, that depends then on, in part at least, your population size. So if you look at these king penguins here, found in South Georgia Island near Antarctica, they have these huge populations. And typically, genetic diseases are very rare when you have really large population sizes like this. So the odds that two individuals carrying the recessive trait are usually going to be, in a population like this, very, very low, because the odds of them coming into contact are very, very low. Okay, so when it comes to population genetics and a particular gene causing a lethal disease, negative traits are usually of a recessive type. There are dominant ones, but without certain scenarios, the dominant ones tend to wipe out individuals that have that really quick if they're in fact lethal. So recessive traits, as far as ones that are particularly bad, are usually pretty rare. Now, that being said, the reason I say that carefully is if you just look at recessive traits and dominant traits, something that's dominant or something that is recessive doesn't really have anything to do with how common it is in a population. The dominant and recessive has to do with whether it expresses itself when you have just one copy or two. But that being said, in this particular case, as far as diseases that are particularly genetically related to recessive diseases, the traits that are recessive are usually rare in scenarios like that. Negative traits are typically rare also in a population. And again, that depends on the population size. However, negative traits also tend to run in families. So if you have some negative genetic traits in you, the odds that your close family members have those same genetic traits tend to be high because you're in fact genetically related to them. So that makes sense. So the problem with inbreeding is, in part, if you have two closely related individuals and they have some lethal recessive trait, you often wouldn't know that in one individual or the other because the dominant trait sort of hides that. But if those two individuals get together, the odds that that gets expressed end up being much higher, obviously, right? So that's part of the problem with inbreeding or making babies with close relatives if you're an animal. Okay, well, this exact thing is happening in a small, isolated mountain lion population in the Santa Monica Mountains. So inbreeding in these mountain lions has become a big problem because as the population size gets small, and so what ended up happening in this case, they produced 3 offspring, but because the population size is so low, the individual known as P12, and the P, by the way, if you didn't pick up on this already, the P is the letter that they use that represents the genus of the mountain lion, which is a puma, and then the number is the number of individuals they have radio collared. So anyway, P12, mountain lion P12, ends up breeding twice, and one of his own daughters ends up being the mate in that second round, and they produce the mountain lion cubs known as P23 and P24. And that's when animals reproduce with someone close to them. You end up with these closely related individuals tend to have a much higher probability of having some genetic disease get expressed when that happens. If you look at cheetahs in Africa, the cheetah population has crashed a long time ago in an experience that we call a bottleneck. And what happens with a bottleneck is you have a very large population that gets wiped out for one reason or another, or some event like that happens, so that your population gets dwindled down to a small number, and then that small number reproduces, and you end up with a lot of individuals again. But the problem is genetically, they look like a very small population because the bottleneck cheetahs has led to extreme inbreeding of cheetah populations. So they're genetically so similar that if you were to try to take a piece of skin from a cheetah and do a skin graft onto another cheetah, there's no tissue rejection. What ends up happening with cheetahs then, in the case of inbreeding, is you end up with some cheetahs that have abnormal sperm. The sperm might have two heads on it, or it might have two tails or some other kind of issue like that. And of course, negative traits tend to be expressed with inbreeding, and you end up getting high juvenile mortality in that particular case. So inbreeding increases the probability of unfavorable traits being expressed, and the negative traits are usually recessive. And again, with recessive traits, the only time they're expressed is when an individual is homozygous recessive. So in summary, inbreeding tends to fix undesirable traits. And by fix here, this is very important. We don't mean make it better. Instead, the word fix means here to sort of solidify it or make it a common thing that continues to reoccur in a population. So in the case of cheetahs, high juvenile mortality is occurring, and that's also associated with inbreeding. And inbreeding results in the loss of genetic variation, the loss of what we call heterozygosity. Inbreeding also reduces a population's chance of surviving environmental change. Genetic diversity can be seen at many different levels. The lowest level is if you have two different alleles, and that's kind of what we were just talking about. So if you have an allele A or allele B and it codes for some particular trait, that's the genetic diversity at an allele level. We're going to really focus on in this class is mostly what we call population level of diversity. So we're mostly going to be interested in the population level. We want populations that have a lot of genetic diversity. And we have population, and we also, in that same sense, and we want a lot of populations that have a high degree of what we call heterozygosity, meaning that they have a wide variety of traits. They are not homozygous, just carrying around just the same traits. They have a lot of variation in them. The next important thing is then how big does a population need to be? Well, it depends on if you're talking about a short-term survival, which is usually 100 years or less, or you're talking about a long-term population survival, so greater than 100 years. And those numbers come from mammal populations. So what I want you to focus on here, and what I want you to remember, is that the minimum viable population, that's what we call the MVP, for a short-term population, in order to survive for 100 years or less, you need about 50 individuals. Now, there's, of course, variation in this depending on the individual animal you're talking about and whether they're mammals or some other type of thing. So this is sort of a broad picture to give you a good idea on sort of a basic genetic level, even though there can be a lot of variation in this. So the minimum viable population for long-term survival tends to be you need about 500 animals, kind of like the penguins that I had showed you earlier. So a study done on bighorn sheep populations found that bighorn sheep, if you had populations of about 100 or more, those populations persisted for about 70 years. If the population had fewer than 50 individuals, that population seemed to exist for or hold on for only about 50 years. And the author of this particular study concluded that the reason the population declined was that the genetic deterioration of that population led to its demise. Okay, so in summary, genetic variability is important for long-term survival of wildlife populations. Genetic variability gives the population resistance to disease, and genetic variability gives a population the ability to respond to environmental change. And that's a buzzword that's associated with genetic variability, and we often will call that hybrid vigor. So again, you want a large population size because you want to preserve that genetic variability and you want to prevent inbreeding. Okay, so I think that's it for tonight. Hope you're all having a good day. Talk with you all